Thank you very much, and now it's my great pleasure to have Steve Joffe come by, good friend of mine, Associate Professor in Medical Ethics and Health Policy at the University of Pennsylvania. Thanks, Steve.
Thanks, Becky. It's really great to be here, and I'm really sorry I missed yesterday. It sounds like a great discussion. I hope that nothing I say proves to be completely wrong, because I missed seeing the light in the discussion yesterday. So I'm going to talk about some ethical considerations as I see it, in seamless drug development. And some of the key points I want to make are really hypotheses, because we're so early in this game that I'm not sure we know how this is going to play out. So these are sort of my thoughts looking forward. Are that it strikes me that these designs, and I think Mark's talk addressed this as well, pose real but manageable risks to study participants. We just have to be aware of them. My own view is that they potentially pose greater risks to the patients in the future whose treatments they influence based on the decisions that are made about drug use and approval. And that in order to minimize these risks and maximize their benefits, we're going to have to be thinking both about the methodological and the operational challenges. And again, I think Mark's talk sort of reflected this. So just as a framework, I think a very useful sort of outline for thinking about the ethics of clinical or human subject research in general and drug development in particular is that it must meet 7 requirements. And so we should think about how these apply in the context of these seamless design. So the first is that the research must have social or scientific value. The second is that it must be designed and carried out in a way that gives it scientific validity. The third is that the subject selection in the research must be fair, that the benefits and burdens of the research and the knowledge are distributed fairly. The fourth is that there must be a favorable risk-benefit ratio, taking into account, on the one hand, obviously the risk to the participants, but the benefits, we're really talking both about potential benefits to the participants as well as the benefits to society or to future patients who are going to gain from the knowledge of the trial. Fifth is independent review. Sixth is informed consent. And finally, a series of things that demonstrate respect for potential and enrolled subjects. This is really a seminal paper from Zeke Emanuel and colleagues from about 15 years ago, and I'd encourage people who are interested in following up on this to take a look at that paper. So here, in this context, looking at these 7 requirements, I think we're talking about 3 principles or 3 criteria that apply in this context, but also 2 classes of patients. So the first principle that we're going to have to be thinking about with respect to ethics here is the question of social value of this research. And I think few of us doubt that the social value of doing research that leads to evidence about effective and safe use of drugs for cancer patients is highly valuable. Second is the scientific validity of the designs and the studies that we are using. And the third is favorable risk-benefit balance, taking into account both risks and benefits for the study participants, but also the benefits to those whose treatments will be affected. So we're really talking about 2 classes of patients. First, the future beneficiaries, where it's they who are affected by the social value of the research, the scientific validity of the research, and they are part of this risk-benefit calculus. And second, the trial participants, and for them, we're really talking about the risks and benefits to them of participating in the studies as research subjects. So the overarching goal of the drug development process is to provide evidence of safety and efficacy so that physicians and patients can make good decisions together about how to treat patients. And I hope everybody agrees with that as at least one plausible statement of the goal of drug development. If we want to speed drug development, if we think that we're moving too slowly, I can think of at least 4 ways that we might do that, and you all can probably think of others. And we already do many of these. One is to approve based on surrogate markers rather than the harder evidence of survival or quality of life, definitive quality of life benefits that you might see that are really clinical benefit. A second would be to skip controlled trials and make decisions based upon single-arm studies. A third is to accept lower levels of statistical precision, to say, well, we're asking too much precision of our current designs or approach, so let's accept less. And then most radically, and I'll come back to this in a few slides, to say, well, let's not even worry about efficacy, at least in drug development decisions. Let's just require evidence of safety. And personal opinion here, and we can talk more about this in the discussion, this is an incoherent position. But it's one that, as you'll see, has been advanced, including by prominent members of our community. So there's a couple of rationales. If you think that we ought to speed the drug development process, there are a couple of rationales that you might advance. One of these is that there are some requirements of this traditional process that we're all familiar with that add little or no value in terms of the evidence about safety and/or efficacy. So you might hold that position, and then we'd want to talk about what is the evidence that you have to suggest that that is true. Another is more of a trade-off perspective, which is we can actually accept lower levels of evidence about safety and/or efficacy when we're approving drugs. This acknowledges that there is a trade-off between, on the one hand, careful and rigorous evaluation, on the other hand, speed of time to approval and clinical use. Many in our community and in many communities have advocated for speed, but I have yet to see anybody really articulate which of these two rationales they hold to, and if the rationale that they hold to is the first, what's the evidence that this is true, and if the rationale is the second, to really be upfront and explicit about these trade-offs. Now, these seamless designs are not, they're moving us in a particular direction, but they're not completely new. So our traditional sort of step-by-step approach of phase 1, phase 2, and phase 3 you're all familiar with. But then there's these hybrids, right? There's phase 1, 2 studies where the phase 2 is pre-specified at the time of phase 1 or at the time of approval of the phase 1 design if certain benchmarks are met. Statistical properties are worked out, et cetera. And then there's phase 2, 3 designs where either with a single arm or randomized phase 2 trial may move directly into a phase 3 trial, again, if certain criteria are met. So this is a sort of hybrid approach here, and I think provides some precedent for the seamless designs that we're talking about today. But the trials that we're talking about today do raise novel go, no-go challenges, and I think, again, Mark's presentation nicely showed this. These traditional designs have explicit pre-specified criteria for phase transitions. They involve statistical justifications that are worked out, and they are subject to independent prior review, scientific review, IRB review, et cetera, both for the benefit-risk considerations and for the scientific validity considerations that are so important. Seamless designs, it strikes me, and there's more expertise in the room than I have, but it strikes me based on my reading of this, that the decisions are much more real-time, ad hoc, and happen on really short timelines. And it seems to me that the statistical justifications and prior approval are much more challenging for the seamless designs than for these prior hybrid phase 1, 2, or phase 2, 3 designs. Now, of course, they have both upsides and downsides, and that's the reason we're all gathered for a day and a half. Potential upside is that they might avoid unnecessary delays in demonstrating the safety and efficacy of novel agents, so that these novel agents get to patients more quickly. Their potential downsides, conversely, are that they might increase risk to trial participants due to the more rapid evaluation of safety and ultimately of efficacy that they involve. And then they might increase risk to the future patients and incidentally, or maybe not incidentally, to all of our wallets due to the approval of unsafe or ineffective agents that are nonetheless going to be sold at a very high price. So a couple of notes on risk for participants. And as I said at the beginning, I think that these are manageable and there are trade-offs here and we can accept those trade-offs. We just have to acknowledge them. So this is just one example of how speeding the drug development process, this here not seamless designs, not the full drug development pathway, but in phase 1, I sort of grew up believing that we can accelerate phase 1 designs and get to sort of quicker decisions about safety and move on to phase 2 and phase 3 more quickly based upon novel statistical designs. This was the first effort that I'm aware of to look at this systematically, looking at a large number of phase 1 oncology trials and comparing different designs, some accelerated, some more traditional, and asking, do the accelerated designs get you to your answer more quickly, and if so, is there greater risk to the study participants or not? And the short answer to it is that the aggressive dose escalation approaches were associated with, in my view, modestly increased rates of hematologic and non-hematologic toxicity. But the key thing is that the response rates, which was the efficacy argument for doing these in the first place, were not improved. So here's some empirical evidence that if we study these trials carefully, we can get some sense of what the trade-offs are in doing this. In this case, it was actually hard to make the argument that these accelerated designs were better than traditional designs. The other thing many of you may have seen in the the Times just over the last few days this article about the toxicities of immunotherapies with respect to autoimmune, including life-threatening autoimmune reactions, new onset of type 1 diabetes in older patients, et cetera. And one of the things that happens as we move more quickly and as we have smaller numbers of patients involved in our drug development programs is we're going to be much more reliant on the post-marketing period to get a sense of what the safety data are. This was a gentleman who was on a clinical trial of, I think, nivolumab and ipilimumab for advanced melanoma and almost died of an overwhelming autoimmune reaction. The other thing about safety here is that these trials are really going to challenge IRB's oversight function. And you can imagine that from the timelines that Mark showed. So from a regulatory point of view, IRBs must find that, quoting from the FDA's version of the common rule, risks to subjects are reasonable in relation to the anticipated benefits, if any, to subjects, and the importance of the knowledge that may be expected to result. Now, as we have these very rapid protocol modifications and seamless designs, IRBs are going to be challenged to do this. I think we're going to be hearing about the DSMB function in a few minutes. That DSMB equivalent for these trials might perform this role, but I think that IRBs still at the moment have the regulatory responsibility to make this determination, both for initial approvals of studies and for protocol modifications. So you can't just delegate it to a DSMB. So I think we're going to need highly specialized, highly nimble central IRBs to make this a practicality. Now, I also want to talk about the considerations for the patient beneficiaries, the patients whose treatment decisions are going to depend upon the results of these trials. And here's where I actually have my greatest concerns. So the context here is that there's this intense public, political, and industry pressure for speed and access. Obviously not an oncology indication, but the Sarepta controversy, which hopefully everybody's familiar with. These are a couple of boys with muscular dystrophy testifying at the advisory committee meeting in favor of the approval of Sarepta, a drug that was ultimately approved based on the thinnest of possible evidence. And from the article in Stat News, the fate of Sarepta has been closely watched as a litmus test for an intensifying struggle between the FDA and patient groups that want the agency to take a more expansive view towards approving medicines for unmet medical needs. That's the context of the pressure. Here, Andy von Eschenbach, former FDA commissioner, NCI director, FDA should approve drugs based on safety and leave efficacy testing for post-marketing studies. Here, Joseph Golfo, writing an editorial in the Wall Street Journal, speed drug approval by getting the FDA to rate for safety and efficacy instead of long-term outcome. I'm not sure I understand the distinction between efficacy and long-term outcome, but that's, there you have it. First change involves returning the FDA to its original role under the law. That is to prevent snake oil from getting on the market by ensuring that the only drugs approved for sale have demonstrated biological activity in fighting a disease and can be labeled for safe use. Biological activity, right? Not surrogate endpoints, not clinical endpoints. The FDA would no longer require approvals based on long-term health outcomes. Here's Vince DeVita, former NCI director, luminary in our world, writing in The Death of Cancer. DeVita does not like the FDA. When it comes to cancer, the FDA has reached far beyond its central responsibility, assuring the safety of new drugs. Might want to put efficacy back on that list. And then quoting further from this chapter, a legion of, quote, Dr. No's has been created, and they're particularly prone to saying no to cancer drugs. That's very bad news for cancer patients. So pressure coming from within our community. And finally, of course, 21st century cures, approved by, well, signed into law within hours. And here again from Stat News, which by the way is a fantastic source of information about health policy developments. The Cures Act includes numerous provisions that set the stage for the faster approval of prescription drugs and medical devices. Although proponents claim these new measures will not lower safety standards, numerous critics, including some former FDA officials and national consumer groups, disagree. They've argued that FDA already moves faster than similar agencies in other countries. Our community lined up behind this bill. I think many of the people and organizations in this room lined up behind this bill. I'm really worried about the impact of this bill on the FDA, and that we may have paid a very high price for that. Finally, coming back to these seamless designs, I want to suggest that it actually is possible, and again, we're at early days of doing this, but it's possible to design trials with contingent and pre-specified modifications, kind of more sophisticated versions of the phase 1/2s or phase 2/3s that we've been talking about. This is an article struggling with how to design trials for Ebola therapies. You can imagine the sort of time pressure, severity of illness, all of that for Ebola, struggles about whether to use placebo-controlled trials, whether to use randomized trials. And this is a nicely worked out statistical modeling exercise of how you might design a trial to get you to the right answer as quickly as possible. And I'll just walk you through the kind of key figure from the paper. The model that they have, which they work out the statistical properties, and they turn out to be quite favorable, especially if there are either very large or non-existent or very small effects from the investigational agent, or to start with a phase 2 single-arm study. This is not starting at phase 1, but you can imagine the kind of phase 1 steps before this. Start with a small phase 2 single-arm study. If it appears that the effects of the drug are very large in that phase 2 single-arm study, then you move on to a phase 3 single-arm study, not a randomized trial. And if that seems to confirm what you've seen in phase 2, then you roll out the drug, you approve the drug, and put it into practice. If phase 3 looks more modest than you had thought based on your early phase 2 data, then you switch your phase 3 trial into a randomized trial where you can really work out whether there's real effects as compared to some sort of control. And then based on the results of this traditional randomized trial, you make a decision about whether to approve or reject the drug. If here it looks like the effects are really quite modest, then you move directly into the randomized trial, which is what you're going to need to demonstrate modest effects. And if it looks like the effects are very weak, then you just reject the drug and move on to something else. So these are the sorts of trials that you can do, that you can model, that you can pre-specify all the decision rules along the way. It also points out that effect size matters. So if we're talking about something with a very large effect size, then a seamless design, I think, is unlikely to lead you to the wrong decision. But if you're talking about something that has a small to moderate effect size, then I think that there's a very high risk of mistaken decisions based upon seamless designs. And we have to be very careful about this. And if and when these designs come into more common use, there's going to be pressure to use them in smaller and more moderate effect sizes, and that's going to be a problem. In fact, that's, I think, the greatest threat that they hold. So the designs and the decisions that are made along the way really have to be contingent on the effect sizes that seem to be emerging at the various stages of the pathway. So finally, I want to offer 4 or 5 recommendations for how I think we can use these designs in ways that are safe both for the study participants and also for the patients who are going to benefit from them. The first is, I think there's a lot of statistical modeling work, and maybe this has started and maybe some of it's published and I just haven't seen it, to model the properties and the performance of various designs under various sets of assumptions. This is work that statisticians do, and I think it's going to be very important. The second is I'd support the establishment of a registry so that we can track the real-world performance and learn from the experience with these designs as we move forward. You saw that Koifman paper that I showed you on phase 1 accelerated designs and how they played out in practice. Let's have registries so we can see how these designs, if they're going to come into common use, play out in practice. The third is, I think it's going to be very important to assign review to central IRBs that have both the methodological expertise and the nimbleness to be able to respond and review these protocol modifications very rapidly. A fifth is DSMBs are going to play a key function in this picture, and there's going to need to be a very kind of close coordination between the DSMBs and the IRBs. And finally, let's not forget randomization unless the early data suggests just very large effect sizes in the trials, because if we don't randomize, I think we're going to find ourselves making some very flawed decisions. So I will stop there and thank you for your attention and look forward to the discussion.